turning over every couple of years, maybe a few years. But if, DNA is pretty delicate, it's a delicate material actually, but if conditions are good, it's not chemically harsh or too hot, for example, or a lot of oxygen, DNA can last for thousands of years, right? And so this is where we get into ancient DNA. You've probably heard of ancient DNA. That is basically just old DNA. That's DNA that, that someone unearthed in some sample, it could be like a woolly mammoth sample from, 15, 20,000 years ago, and it's still good. That's just an example of DNA found in the environment. Okay, so how about barcode? We're gonna mention barcode and marker. What is that? Well, it's basically, it's a short sequence of DNA or RNA, usually in the neighborhood of hundreds of base pairs, okay? And this is a part of the genome, a sequence area of the genome that mutates just at the right speed over evolutionary time, so that if you look at that part of organisms' DNA or RNA, you can get a specific signature of that species, okay? So each species has its own genetic barcode, basically, and there are certain parts in the genome where it's sort of the Goldilocks principle. The mutation has been just slow enough, not too fast, that area of the genome varies just enough so that you can tell what species you're looking at if you sequence that part of the genome. There are lots of words applied that mean the same thing. Primers or markers or genes are often applied to this technology, but they're all really, they can all be barcodes, basically. Okay, so I wanted to introduce the Cali DNA system and how it works. Essentially, you can go online and you can register. You can sign up for these bio blitzes, these community science bio blitzes, these volunteer events, where you can collect samples and get involved with that science. And then those samples go to labs, they get analyzed, and then these results ultimately get put onto the web, which the idea is that, the public can explore them over time, and scientists can use them to write new studies and figure out new things and write conservation plans, etc. So here's like a little short intro video that is silent, so we can watch in a second here. Okay, so that kind of in a nutshell is how it works if you follow that. But there's a routine, there's a protocol to basically collect samples. Those samples get analyzed, and then those data get used for all kinds of purposes, I said. So I invite you to go onto the California eDNA website. You can join it, you can sign up, you can see what events may be happening, you can explore the projects that are currently listed. There are a lot of projects, and I'll go through a few projects here now to give you a sense of the flavor, but it's mainly driven by volunteers. And hopefully this is because of that volunteer effort. Hopefully it's something that remains, perpetual in our state. Everything's about funding too, though, isn't it? So hopefully that can be maintained in the future, long term. Okay, so I'll showcase a few uses or examples of this project. Here's an example of California coastline sediments being sampled. This was organized by Mike Dawson at UC Merced. But typically what happens is a BioBlitz eDNA survey event is created and on a certain date at a certain place. And citizen scientists or community scientists are recruited on that day, and everyone meets out there, at the beach, and they're given their materials. usually organized by someone, often Rachel Meyer is there, to be honest. I don't know how she gets everywhere, but she does. And then, you know, so those are really cool, fun events. And then on the side, there are courses and modules that take place at different places, often at schools or universities, and students are involved. And data sharing is, you know, is just throughout the entire process. Okay, and so what are we looking for? What are some of the goals of this work? Well, we want to go from this, where, you know, we arrive at a place and we just have a list of the species, which is great. It's great to have a list of the species, don't get me wrong. But usually, you know, what we are most aware of are the macro species, you know, like the larger animals and plants. So with eDNA and these barcodes, we can get everything. We can understand the entire spectrum of life that is detected in a sample. And as you can guess, most of the diversity, or know already, most of the diversity of life is at the micro scale. Okay, so a lot of what we are seeing in these samples are undiscovered organisms or species. That's probably the majority, and that's exciting. But you can, you know, you can take these data over time, and you can make biodiversity maps and understand biodiversity from a large scale. You can sort of organize data to understand, you know, which plant groups, for example, are found at which locations and what the patterns might be. Over time, you can create what we call network analyses to understand how important species interactions might be, just based on presence and absences, and see how many times and how often certain microbial species are found with larger macro species, for example. And then later, you know, communities can be organized and visualized, you know, in all kinds of cool statistical ways to sort of analyze them and tell how different they are and how similar they are. But there's just so much that can be done with these types of data. And so, you know, the general idea, though, is that there's this dynamic metadata program where you're collecting certain types of data, you're bringing it all together, you have a standardized method, you use some standardized tools for data collection, and then sort of an information pipeline has been developed and is being used, where sample data are processed, the DNA is collected and analyzed. Those data are basically pushed into an information or informatics pipeline that have been, you know, home style created, right, with their own sort of, with our own scientists in this network. And then out pops a lot of standardized information that can be viewed to put data at your local ecosystem in context with other ecosystems across our state or the world. Okay, and I mentioned these primers, or these barcodes, and it turns out that some barcodes are better for some organisms, right? So this CalEDNA project has used 5 barcodes. They're sort of shown here, they're kind of cryptic, but right here, the 16S and 18S. Anyway, they're shown here, and you can see basically the types of organisms that these 5 barcodes specialize for, and there's some overlap, but there's one that's good for bacteria and other microbes, plants and algae, fungi, animals plus, I say, because this is, you know, this marker is good for animals plus other things, and then just a general good eukaryote, which is, you know, more complex organisms than bacteria. So we're using a whole, you know, set of barcodes so that there's a lot of overlap, and you are less likely to miss organisms in the environment. Okay, and just showing the faces of some other project. There's a cool tide pools project in collaboration by the Natural History Museum of LA and Cal Academy of Sciences. That's really cool. And so, I mean, what's not great about hanging out in tide pools? There's an example of CalEDNA being used in the Santa Monica Mountains to use for post-fire community reassembly, to understand basically mainly what was there before the fire, and by looking at unburned areas and what is there after the fire and how that's changed. not only the microbial community, but perhaps, the seed bank as well. And we're still just learning like how resilient DNA is to things like fire. So, if you can detect an organism in an ecosystem before it burns, can you still detect that organism and ecosystem after it burns? So, just learning a lot of things like that. We're still in the baby phase for a lot of this, of understanding like, how easily this DNA information disperses in the natural world and how long it persists. But it's a hugely burgeoning field right now of research. Okay. Here's an example of Pillar Point on the West Coast, where you can compare eDNA samples, right, taken from just visual database samples, right, from users over time who've recorded different species. At this point, you can take data from eDNA, you can compare it to public repositories of data, and then sort of see what the diversity picture looks like when you put them together. So that would be this eDNA picture versus GBIF. And there's surprising results sometimes. There's different, you might get much larger spikes of diversity in, for example, in eDNA, you detect higher diversity in this unprotected embayment in this Pillar Point, whereas, it looks like a smaller signature when it's just, observed species out there in Pillar Point. So there's a lot of, cool ways to overlap different types of data and see how they compare to this eDNA method. You can detect, I mentioned biological networks. You can detect, the number of species that show up together in samples, and do lots of cool and interesting network analyses, and build these, these community models potentially of species interactions. Something that, if you were just collecting a soil sample, preserving it, putting it under a microscope, let's say, and seeing how many, things you can grow from that sample or detect with a microscope, it would take ages, right? So this is a really cool way to see how many things are in one sample and how well they relate to one another over time. eDNA is being applied to desert systems, which are vastly understudied in general. Here's an example of using crayfish, for example. I think my internet got a little unstable there. So there's invasive species and detecting invasive species, or really rare, threatened and endangered species in important protected environments. That's another important use of eDNA. And Denise Dr. Ruiz Ramos, who worked in our lab for a couple of years, that's what she does in the Missouri River now for USGS. She uses eDNA to detect aquatic invasions in our largest trade systems or waterways in the country. Okay, and then just as sort of an information tool, eDNA is super handy. These are different traditional maps for understanding biodiversity. For example, this is sort of an ecosystem or biogeographic approach for understanding an ecosystem. On the lower right, you have the USDA ecoregion system and the EPA ecoregion system. But if you take environmental DNA and that complex diversity and you map it at that fine resolution, you can get maps that look like this that tell you about hotspots of turnover. And it turns out the Central Valley and coastal regions of California are actually, even though the Central Valley is not considered a huge biodiversity hotspot relative to other parts of California, it is a place of huge biological turnover. So lots of eDNA, lots of DNA is moving through this valley. As you move from one mile down the road to the next, you are passing through a ton of biological turnover and change. So anyway, maps like this can be produced and this information shared. And this is our own project at UC Merced, where we're using eDNA to understand vernal pools better. And we've started off by sampling a set of vernal pools, and we will sample the bottom, the edge environments, and the upland environments of vernal pools, and ask, at different time points and in different pools and in different seasons, how does the DNA turnover change, basically. And I'm just showing you a glimpse of some initial data. I know this looks kind of complex, and it is, but these are these sunburst charts. And so I'm looking at a sunburst chart for this one marker, ITS2, which is a plant and algae-based marker. And this other marker, CO1, that covers a whole wide range of organisms. But these are the kind of data that get produced from a sample by understanding all the species detected by one marker or two markers. For example, you can display this huge array of diversity in visual form. In this case, one of the things we're interested in is detecting these threatened and endangered vernal pool plants, like this calusa grass, Neostaphyia calusana. And you can see this arrow is pointing right here to the sequence that was detected, which shows calusa grass in this vernal pool sample. So once you have a nice reference database built up, if you apply these barcode markers, you can detect hundreds of species in one little pinch of soil. And this figure, the whole takeaway here, we have five markers shown here. It's just a reminder that we're using 5 barcodes. But you're seeing like 2 time points of sampling in the different colors. So red represents collections from 2017, blue triangles represents collections from 2018, which was a drier year. And that sort of goes to show the variation that you're going to see, not only in space, because we have bottom environments, edge environments, and upland environments for these vernal pools. You can see that, for all these markers, there's some variation between these zones of the vernal pools, but we see big variations in different years, right? And what variations in what? I didn't tell you. This is diversity. So the number of species detected by these different markers is shown here in the y-axis. And on the x-axis, we see the different habitats within these vernal pools. And so the takeaway here, I said that eDNA can last for years. It does, but the turnover can also be great, just even between 2 years that are right next to each other. So it's very dynamic. It's very dynamic, and we're still trying to understand that dynamism and how to leverage it to give us a really clear and powerful view of these ecosystems. Okay, so that was the eDNA part. I'm going to shift to the California Conservation Genomics Project, which will be shorter. But I want to acknowledge a couple of people in particular. These are folks who have worked really hard on this project, but in particular, Brad Schaefer, I showed a picture of at UCLA, Professor Brad Schaefer, and Mark Gold at the California Natural Resources Agency were pivotal in basically wrenching $12 million from our state government, convincing legislators to let it go, and the governor to take a chance on this genomics projects, which I'll tell you a bit about. Okay, well, first I want to show you this slide. Let's take a couple minutes on it. All right, what do we get when we look at the entire genome of an organism? What information can we get out of it related to conservation? So this is a really cool, interesting paper. The genomic signals of selection predict climate-driven population declines in a migratory bird. So this is a yellow warbler here. This is a super cool study first authored by Rachel Bay, who's a professor at UC Davis. But what we see here mapped onto North America, or at least northern US and Canada, is a map of genomic vulnerability. What the heck is that? So darker red areas or darker areas are areas of higher genomic vulnerability. Now, what that is that the genomes of these birds were sequenced across their species range. And the genetics was correlated with the climates across this species range in this continent, right? And in areas where, you know, you get certain genes being adapted, right, and being fixed, in areas where those are related to certain climates, you'll expect that there's going to be kind of a mismatch between the genes you find, like let's say there's some genes that are highly adaptive for the Rocky Mountains, right? But in the future, these environments are going to change and move, right, with climate change. And so the genes that are most mismatched, the adaptive genes that are most mismatched with future climates are what are mapped here. Okay, so when you have big mismatch between genes that are found in current climates and what those climates will be in the future, we call that genomic vulnerability, meaning like you have genes in the wrong place for what's going to be coming down the road. Okay, that's essentially what that means. What this study shows is that places in the species range where there's population declines, that's also shown as dark colors, right? Those are places of highest genomic vulnerability. Okay, so basically, populations that are mismatched with, you know, a warming world are already seeing big population declines. That's the take-home message. That is just an illustration of what can be done with genomics, and that's something that we hope to do with this California Genomics project. Okay, so let's just run through it, the attributes of this project. So the goal is to sequence the genomes of about 225 species, about 130 projects total. And the strategy is to create a reference genome. That means like sequencing the genome for the first time of an organism, right? Remember the human genome was sequenced, it took forever, the Human Genome Project. We can do it now like in an afternoon. We can sequence a genome of an organism. And then what we do is we collect individuals across the species range, you know, around 150 is the idea, individuals across the species range, and you do what you call resequencing. And that means you sequence them again and you compare those sequences to the reference genome, the first genome that you sequenced, okay? So basically doing this for 225 species at about 150 individuals per species gets you about, you know, upwards of almost 20,000 total genomes resequenced. It's a lot of data, okay? And to do this, they've created a dedicated core facility at UCLA. They've created a uniform genomic coverage standard. That's jargon we can talk about later if you want. And they've created a common analysis pipeline to make, you know, combinability of the data more possible. Okay, what do we want to accomplish? Okay, so we want to accomplish taxonomic and ecological coverage for the state replicated across many species. I mean, there's thousands and thousands of species in our state. So this is scratching the surface, right? Just the beginning. But we want to also develop meaningful maps of variation across the state so we can, you know, identify hotspots of genomic variation that are climate resilient. Corridors to connect these hotspots, right? And then also just critical lands, habitats, regions, etc., to acquire and protect, or, you know, create partnerships to protect. Who's doing this work? So right now there are about 71 principal investigators. All 10 UC campuses are involved. There's an executive committee. The governor's office is involved. U.S. Fish and Wildlife Service, California's division or Department of Fish and Wildlife Service, several hundred co-principal investigators, and the California Department of Food and Ag is involved to, you know, to leverage some of this funding for pest and agricultural species of interest as well. Okay, the funding, $12 million from the state budget, about 10 million of it goes directly out to the labs to do the work. All data must be consistent. All reference genomes are done within this project. All resequencing is done to maximize landscape coverage across the state. And most of these projects are related to conservation sensitive species. That was a hugely important thing for Brad Schaefer. who's a conservation biologist, loves herps. He loves turtles and frogs and toads, but he loves all biodiversity. So he wants to conserve things. This is what started him off on this project. So just to show you the coverage from this project, these are marine samples that are being acquired, plant samples across the state, vertebrate samples, and invertebrate samples. So, it's fairly wide coverage, and that was the whole goal. That was what the team was trying to accomplish. With the sampling that is ongoing, you can see where you have high, average, and low number of species representing different ecosystems. The Sierra are well represented in the study, as well as a lot of coastal regions. The worst represented regions are, as often is the case, like deserts, southeastern deserts, northeastern desert regions, and eastern desert regions, right? And some of, the northern areas where there are lower populations of humans, right? Three cool things in the works, okay? So one, construct a statewide guide to hotspots of genomic resilience. Two, enable some forensics or repatriation where it might be needed, places where populations have been lost and need to be restored. And ultimately to provide a lasting resource, and that's the real trick. CCGP goal is to create a resource of whole genome data, publicly available and easily queryable for all users, meaning it's a resource out there, it's completely public, anyone can go view it, use it, and forever. To combine these data with geospatial data layers, so you can, answer all the questions that you want to. The key problem, of course, is making that resource long-lasting and available, and it's really impossible basically without long-term support and funding. So that's always, that's the crux. That's the human condition though, isn't it? That's everyone struggles with that problem. So that's just being upfront about it. Okay, and then so there's this CCGP website that I encourage you to go explore. And let's see, I think I can go explore it right now for a second. And when you get there, you can read more about it than I'm blabbing on about. You can see some cool tools and potential genomic maps. I think the funnest thing is to go shop around and look at the species that are involved in this project. And so you can do that. There are, like I said, there are over 200 species. You can click on any given species, and there's a little write-up about that species and why it was selected. Usually it has some interesting conservation importance. Okay, so I invite you to go do that, check that out. All right. Okay, so I'm gonna just finish off here with just highlighting our own projects for the California Conservation Genomics Project. So I mentioned vernal pools. We have the gorgeous slide, I think it's beautiful, but my wife took it, of the Merced Vernal Pool Grassland Reserve. And here's another picture of our reserve in the summer, right? So vernal pools, they're ecosystems that are, mostly active in spring. They're vernal pools, spring pools. But as the seasons change, even in mid to late summer, you get these really, interesting adapted vernal pool specialist species like this Calusa grass, this Neostapia calusana shown here in Daniel Tay's hand, who's a grad student, one of my graduate students. This is sort of the magic and the wonder, but the sort of endangerment of these systems too. They're 95 to 99% destroyed in California, but they're hyper diverse and they're extremely special and interesting. And so Lily Pennington, who you'll hear from next month on monkey flowers, she's actually studying these grasses as part of her dissertation, as part of, with support from this California Conservation Genomics project. So next generation of scientists are doing this, this challenging but cool genomics work. And so, she's focusing on the conservation genomics of these ore-cut grasses, doing whole genome sequencing, marker development for this clade to be able to detect it in other pools across the state. and looking at whole range assessments of genetic variation so we can understand, again, where are the remaining hotspots. Pretty much we have to protect about everything that's vernal pool these days, but we still can learn like where are the biggest hotspots of genetic variation. And then a second part we have in this project is monkey flowers. I often study monkey flowers. And for the Conservation Genomics project, we're resequencing the genomes of a widespread species in the state that I'm sure you're aware of, the common yellow monkey flower, but also a Sierran endemic, the cut leaf monkey flower, which is only found in the west slope of the Sierra. So we'll be looking at, the genomic reach of these species and compare what, common and widespread species, what they look like in terms of their genomes. Okay, that's it. I wanted to thank a lot of funding bodies for this work, and I want to give you some time for questions in case we have a bit of time. So thank you so much for your attention. And is anyone out there still? It's a question too.
Yeah, we're all out here. Thank you so much, Jay.
Oh, pleasure. Thank you.
Does anyone have any questions?
Go Jacks, go Ags. I'm looking at the chat now.
Oh, yeah.
Mm-hmm. Yeah. And yeah, Tom Hodham's, yep, on there. Yep, you're right. It's a dry year, Tom. It's but it's still gorgeous right now. It's just still beautiful. It's not as wet as those pictures.
Hey Jay, how could Sierra Streams Institute scientists or citizen scientists help with the CCGP project?
Yeah, thank you for that question. Yeah, so keep asking and I will get back to you, but one of the most obvious ways, which may not be as satisfying for perhaps, is to help these scientists actually detect and collect their species. So these study species, which I mentioned, a lot of them are kind of in hand and have been collected, but a lot of others are not. Like, for example, we're still looking to collect for our study species. So, I mean, one thing anyone can do is go onto the site and sort of go, see what species are there and see if like you have those species around you. And if you contact CCGP, they would love to get information about where you're seeing things, and perhaps you could help collect them. You could also, I would say, by sort of digging into and learning more about the project, or reading about the project, you could certainly influence the project. If it has aspects that you think are really relevant and important, I would say contact CCGP, contact me, contact Brad Schaefer. We love involvement from organizations such as yours, and you have your eyes on this region and many other regions, I know, so you're an important voice. But it's a little tricky. I was kind of semi-anticipating that question, but it's a little harder for me to come up with that. If I was Brad, I would have more brilliant ways to tell you right now, but the Cali DNA project has more obvious ways for interaction, but the CCGP has been a lot more sort of top down and sort of going out, sort of finding experts, getting recommendations of species to study. But I think that we're in the middle of this project, and it's not too late at all to influence which species are included. So if you have, I don't want to say pet species, but like if you have species of importance that you think could really lend an important view to conservation genomics across the state. And that's the other thing, we're trying to find species that do have a wide range of environments, even if they're threatened or rare. And that's challenging because a lot of our threatened and endangered species are only found in a few spots. Yeah, I'll stop there, but that's one long-winded response to that quick question. Thank you.
Thank you.
Yeah.
I think there's more in the chat.
Yeah, so I can go into those. What is the cost of one test, right? So that is the bad news, right? So it depends on if we're talking about, and Paul submitted that question. If we're talking about a sample, like an eDNA sample, for example, you know, it can range, you know, within dozens of dollars to up to $100 to actually get that sample completely finished all the way to the end where we have the data in hand and we're looking at the genetics data. But it just depends on, you know, what we're talking about exactly. So it's not cheap. And so I should say too, you know, the eDNA project is collecting a lot more samples than it can process, right? So the idea is that we collect samples, they're kept, they're held, they're saved, and then they're looked at later as other sort of funds become available. So right now, a lot of it is in an archiving stage, with smaller projects being, you know, completely analyzed. Will this project assist adaptation to global warming? I think it can. I mean, certainly that is certainly the goal. And, you know, sort of like I mentioned, one way to do that is you have to identify populations that are in the biggest trouble, and you have to identify populations that are most rich in adaptive genes. Okay, so to really, and it's not just a species process, it's a regional process too. And I probably skipped over that idea. And that big map I showed where I was overlaying the plant and the animal, the marine, the invertebrate, you know, parts of the state, we really hope to develop a complex, you know, multiple holistic picture of parts of the state that are in higher genomic trouble, and also have higher genomic resilience, you know, where there's a lot more variation involved. As you know, our state is just, I mean, it's so diverse, it's so incredible, it's so huge, and it's so changing. It is so dynamic, you know, so, you know, something that may have happened 30 years ago, the signature may still be there, but also the other thing I want to tell people, which you, I'm sure, appreciate a lot, is that restoration is so possible, you know, in ecosystems. You know, it doesn't have to be a perfect return to the past, but genetic, restoration in terms of restoring diversity is probably going to be the biggest trick we have in combating, or future generations have in combating, this climate change mess we've gotten ourselves into. How can we contribute existing DNA sampling results? Conversely, participate in sampling for genomic resequencing. Great question. I mean, one of the tricks for that CCGP project is to try to create a standardized set of data and reference data. So that might be possible depending on what we have, but most of these species were chosen, if not all of them, because they don't have a great reference genome already. So we expect a lot of the data generated from this to be new, but if you have collections in hand, maybe DNA in hand, that could, I mean, I don't see why it couldn't be helpful. So I'd say contact us. Okay, in your opinion, do you see eDNA collections replacing traditional surveys eventually? No, not at all, 100% no. And that is, I think, partly because, like I said about the eDNA lasting in the environment, in the Calusa grass example that I showed you, I showed you that vernal pool, which had probably hundreds of thousands of individuals of grasses in it, right, that emerged. We only detected that species DNA in one of the years that we sampled. The other year we sampled that pool, the dry year, we did not detect it in the soils. And that was like 9 scoops of the little scoops of the soil. So we're learning a lot about what it means to get a negative result and what it means to get a positive, and how long it's good for. And so no, it will never replace it in my thinking. So we'll always need naturalists, and we'll always need people with the rulers and the scopes, you know, and all of that. There's a big, nice, beautiful paragraph from Tom. Tom, do you want to speak up? Can you turn your mic on?
Yeah, I really should, because this is too long. So I'm going to state this much more quickly. I tend to find things out by writing. I guess that's why I do writing is my thing. Anyways,
Absolutely.
the question is, has the project evolved at all under the current conditions of social distancing? I know that I'm finding my environmental writing courses to be surprisingly alternatively nicely satisfying in that we get this kind of bigger picture of people's backyards where they live. And
Yeah.
it's more comprehensive in that regard. So I guess I'm just wondering, just simply, has the project changed at all under these conditions?
Yeah, that's such a Tom question. I love that question. Yeah, so we've been in crisis mode, right? You know, like in the past 8, 9, 10 months. And so, yeah, it's changed a lot over time. I feel like we're in this spring awakening. Of course, I hope it's not too fast and we don't ruin everything and, get infected too fast again. But I feel like field work can totally be safe. And if you just follow a few simple rules. And we're starting to see a real resurgence from the research community. Like reserves are starting to get like getting their rosters filled up for the season and for summer. And so I don't know. I guess one response is that I think we appreciate fieldwork more than ever.
Yeah.
Like that's and people are starving for it. I know I am. I'm just like, I can only go to the field.
Yeah.
Yeah.
Well, I know it's kind of in some respects testing the CalNat program, which has always been very site specific, but now
Yeah.
in some ways is responding to this larger call to understand the state and its varied ecosystems in toto.
Right. Absolutely.
Yeah.
Well, thanks. So I see a couple of hands if we have time. I don't know. There's Saul has a raised hand.
Thanks for the talk, Jay. Can you hear
Sure.
me okay?
Absolutely.
Cool. You may have actually kind of answered this question with Alex's question about, replacing traditional surveys, and I know you've touched on this, but just the ability of eDNA to predict abundance and kind of like where that's going for, the ability to be like, how many are out there versus just kind of like presence absence.
Yes. Yeah, there are a lot of really good, thanks for that question. There's a lot of really good studies on that right now because you can basically quantify the number of sequence repeats that are, obtained in a sample and relate it to abundance, but there's problems with that. And that's what these studies are doing. They're trying to understand when you can do that and when you cannot do that. Mostly it fails, I would say, because PCR, the polymerase chain reaction that amplifies DNA so that we can detect it, way over amplifies, and you get false signals of abundance. But there are ways you can standardize things and do that, and people are doing that. Yeah, appreciate that. How about Miroslava?
Yeah, hey Jay. I just wanted to thank you for your talk. I think the last time we spoke was at Blue Oak Ranch Reserve in 2017, a good while ago.
Awesome.
Yeah, I just wanted to chat real quickly. I'm here based out of UCLA for the Cal eDNA program, and I wanted to chat to folks and let you know that we have over 1,000 of our sites all across California, even some outside of the US, with eDNA results online. So you're welcome to click through and explore, the plants, the animals, the bacteria, the flowers that we're able to see from all of our samples, just like Jay mentioned in his talk today. I also wanted to add that if you're interested in collecting samples out either by a local park or someplace you think would have a lot of great biodiversity, like Jay mentioned, we can't guarantee we'll be able to process the samples. But again, the more samples we're able to have and store in our archives, so when researchers are curious about a certain area, they'll be able to use samples that you've collected out in the field. So I definitely want to encourage folks, despite the pandemic putting a halt in in-person events, there's still ways you can get engaged online. And right now we're working on uploading a lot of education material from our undergraduate courses. We have other volunteers who put a tremendous amount of effort and work into explaining eDNA to their students. So again, we want to make sure that, our citizen scientists, you guys are the backbone to the program. We can't be all over California at once, and really, really, really looking forward to being able to host events in person so we can see all of your lovely faces and hear about your observations and hear about, what makes sites exciting for you all to collect. So sorry, I didn't mean to ramble there, but I just wanted to mention, I'll drop the link to the website so you can explore all of our analyzed sites. And if you're interested in taking like a training kit online to learn how to collect kits on your own time, I'll put the link there too. And if you have any questions about getting involved, having students involved, or, trying to stay engaged from the comfort of your own home, you're more than welcome to reach out to Jay, anyone from the Cali DNA program. It's a huge network, and we're really fortunate to be able to have so many diverse and so many experts from so many fields really putting in their effort and time on it. So thank you.
Yeah, and contact Miroslava. I swear I didn't plant her. Thank you so much. That's awesome. Thank you so much. How long do we have? I'm happy to answer questions as long as we're here.
We can keep going.
Cool.
Yeah,
Okay.
we have you booked until tomorrow night, right, Jay? 24-hour marathon.
I'm gonna run to the fridge and I'll be right back, and we'll just keep going. Now Theo has his hand up. If we want to go to him.
Hey, thanks for the talk. This is really interesting.
Sure.
I'm wondering about like signals. Are there false signals between taxa, even distantly related taxa, or is the IGS region so bizarre that you aren't going to get some other region just randomly having the same sequence?
Yeah. No, great. Yeah, thanks. Yes, there are many false positives potentially, and that's part of the research and part of the challenge. What it depends on often is a really good reference database, right? So you have to have a key to go to when you read that sequence to see like what it is, right? But if that reference is not there, or as you said, maybe a close relative is in the reference database, but not the species you actually detected, you're gonna get a false signal. And we're running into this a ton in our like vernal pool study. You'll get a signature from some, like an African species of euphorb or something. It's because that's the closest relative to some actual euphorb that may have shown up in our sample. So, and then there's undescribed things as well, and they may match to some closest taxon, but it's a huge issue. It's going to be ongoing forever. I mean, not ever, but like a long time in the eDNA field of research. Yeah. So, okay. Where else?
Yeah, I had a question too, Jay, but I was going to defer to Inga's question from earlier that was typed in the chat. It's kind of an interesting one.
Okay. Sounds good.
So it says, how do you collect samples for eDNA without getting human DNA all over it, or is that okay?
Yeah, well, yeah, you do try to keep samples as clean and sterile as possible. So there's sort of a protocol to keep things clean, to keep contamination down. We wear gloves, for example. But it's not the end of the world. You are going to get some human DNA in your sample. And so, luckily we're barcoded up. You just don't want your human DNA to swamp the signal of other species. So it is a challenge though. It's a challenge keeping samples clean, but there's a lot of human DNA in soil too, in places where humans are. So yeah, it's a great issue though.
Okay. So I had a question, Jay.
Yeah.
So it's, I guess, kind of backing out from just like the eDNA and looking more at, again, the intent here in terms of conservation genomics. And you had brought up this idea of genomic vulnerability and finding potentially species in locations where there are mismatches between their environment and their current adaptations. And one of the big questions that comes up often in these kinds of projects where we're trying to maybe use that information to then better build in like climate resilience into a population is kind of this, you know, how much is too much? How do we use that information to then actually do things on the ground? So I don't know if you could just kind of speak a little bit to this idea of, like, I know we talked before about like assisted migration and these other things, but how do we then use that genomic information actually in a tangible way to then do some sort of climate resilience project? Do we just like move individuals? Do we move like bird eggs? Like, what's the actual process that you suggest for incorporating this climate change lag into restoration projects, so that if we go out and collect seed from plants, let's say, how do we ensure that we're maybe incorporating this climate change effect into those seed collections?
Yeah. Wow, that was a heck of a question. That's great. Yeah, that's like, you know, more than my career and many others' career. That's awesome. No, but that's an important question. So I kind of view it as it's an endlessly complex landscape and universe. I mean, just even, you know, if you have an acre of land that you're managing, just even visually understanding and managing is complex, and coming up with a plan is complex, depending on what you're managing for. So, you know, of course, I'm not, you know, you knew I wasn't going to have like a lot of specific answers here, but, you know, this hidden information, it's mostly hidden to us, right? I mean, like we've evolved, our society collectively as humans have evolved so quickly in the past couple hundred years. You know, we're sort of destroying things faster than we're discovering them, you know, or rediscovering them, I should say, right? We kind of went through this period of forgetting a lot of things, I think, through the industrial period, right? But we're sort of rediscovering things. And so I think that it is going to be extremely complex. But I will say this, we have to take risks. We have to take conservation risks, I would say, and do sort of management experiments. I mean, you can call it adaptive management, whatever you want. We have to stop, and these are, you know, a lot of these are sort of common answers to these kind of questions, but like we can't look at systems as fixed, you know, like the museum model. You know, we have to look at them as more arenas and sort of take chance. But I think that the opportunity we're presented with now is to is really to uncover more information than has ever been possible. And it's also challenging because how do you actually manage that information too? Like that is a whole other problem, an interesting problem to have. But I, you know, another stock answer, which I think is just true, is that we're never done. It's like, it's complex, challenging, interesting work that's never done. And now we just have more tools. And so whether you're managing an acre or like a whole watershed or like a county or whatever, you know, it takes as many minds and eyes on the problem as possible. So.
Well, and I hate to add on another question. There's another question. Yeah. We're getting pretty extended in time here, and I'm sorry, but so not to like put you on the spot, but I guess my real kind of question there is, what's the fine line between if we know, for example, what maybe a kind of climate change movement distance would be for a plant, like, hey, if we're going to restore this property, we might want to take some seed from X kilometers south. How much of that is too much? Like, do you think we should be doing that, taking risks like you're just saying? Or do you think we should be also more cautious in how we mix our genomes when we plant a restoration site?
Yeah. I like informed approaches that are, people who are thinking about these problems a lot, you know, are putting more nuance on it. And so, you know, one example I like is, you know, if we're talking about a certain species, we can do this with communities too, though. You know, you can think about migrating X proportions of species in a community as well, you know, or you can think about special genes or genotypes. But think about, first of all, understanding and measuring like a historical, you know, what they call dispersal kernel, right? Which is basically like the distribution of variation. So if you're a stand of ponderosa pine, you know, 5% of your genetics is made up of genes that have probably traveled 100 kilometers. This is just me making it up. 20% is from like 10 kilometers away, and then most of it is right there. You know, most of the genes stay local. So applying those kinds of rules will probably avoid trouble, right? So if we don't just only move the hot adapted stuff, you know, we do a distribution of variation, right? We start with local, we move some of the hotter stuff and then a little bit of the really hot stuff, you know, to try to make that a diverse population or community if we're talking about species, you know. So it's not just like that genotype belongs here and this one belongs there. Populations like human populations, we look around, we're diverse, that's when we're stable and awesome and strong. And that's how we need to look at our natural systems.